1 in 10 women are affected by PCOS, which means that it is 6 to 12% of reproductive age females worldwide, and 5 million persons just in the United States of America. This condition presents itself with symptoms such as pelvic pain, multiple follicular cysts, heavy bleeding, hirsutism, infertility, which can all further lead to more chronic conditions such as type 2 diabetes and cardiovascular diseases. Till date, there's no definite etiology about PCOS. So treatments are based on the associated symptoms that patients present with. And number 1, universal treatment, diet and exercise. Number 2, medications, clomiphene citrate, metformin. And number 3, when all fails, laparoscopy, also known as ovarian drilling. In 2004, a company by the name of Novocure established a technique referred to as tumor treated fields. And this technique was a non-invasive technique in which they would use a low-intensity alternating electric field to help inhibit tumor growth. With this, since then, they have been able to conduct all these different trials over here that you see. And till date, they have been FDA approved for the treatment of glioblastoma with their technique. But what has evoked our interest is, you see this particular one here, recurrent ovarian carcinoma. Recurrent ovarian carcinoma is where they use this technique, and we decided if they can use this in a cancerous cell in the ovary, then what if we try this to observe the effect of this electromagnetic radiation on a non-cancerous condition in the ovary? So what we did is that we obtained ethical approval from the University of the West Indies. We got females from dowry rats. We had standard laboratory conditions, and we had 4 major groups. A control group, an estradiol valproate group, which is the group in which we induce polycystic ovaries. That same PCOS group where it is we had exposed to electromagnetic radiation, and we had a pure electromagnetic radiation group. Now, our research was based on monitoring the oestrus cycle, inducing PCO, performing histology, performing immunohistochemistry, and hormone estimation. Now, if you look at this small video here, this will give you so if we look at this video here, this gives you an idea of our exposure system. So the animals were exposed to electromagnetic radiation, and this is how the setup actually looked. They were induced with oral administration of estradiol valproate, and from this, their body weights were taken on a weekly basis. The animals at the end of the exposure period, they were anesthetized and they were euthanized for blood collection, hypothalamus, pituitary, and ovarian collection. Now, this organs that was collected, it was sent for histology, in which they performed the hemotoxin and eosin staining, and this was then further read with a microscope for us to be able to identify the tissue structures. The second thing that we looked at was the hormone estimation. So we collected blood in these tiny little vacutainer tubes, and these were microcentrifuged into a microcentrifuger, in which it is we were able to collect the serum to perform ELISA testing. After, we were able to perform immunohistochemistry, which was a long procedure of several different procedures in immunohistochemistry to be able to help us identify follicle stimulating, which is FSH, and luteinizing hormone estimation or reactive cells. Now, in total, we will have conducted 6 major experiments. The first 2 experiments was a pilot study, and then it was a trial investigation. And this information was presented to the CAS conference in Jamaica, where they were able to help us fill the lacunas in our research so that we can better design our research for further study. Over the years now, what we have been doing is that we have been establishing the effect of electromagnetic radiation on the regular ovary and on a developing cystic ovary. from this, we were able to produce 4 major papers. So all the results that we collected, we took them, we analyzed them using SPSS, and then we sent out 4 papers for publication. Now this paper, which is currently under review, and this is just our graphic, the abstract here, it basically tells us that we looked at the normal rat ovary and the effect of electromagnetic radiation on it. And we were able to observe that there was an increase in the number of developing follicle cells here, secondary follicular development. We also observed blood congestion, and there was an increase in FSH and decrease in LH. We also looked at the morphological structure of each of the follicles as in the different stages of development. Secondly, we published a second paper which has recently been published earlier this year. And in this paper, what we looked at was that the effect of electromagnetic radiation on developing cysts. So what we were able to observe was that there was a decrease when you expose these animals to electromagnetic radiation. There was a decrease in the number of cysts and the sizes of the ovary. Also, throughout the different follicular development stages, we were able to see that there were small, tiny degenerative changes. Additionally, we found that an estriol valvuloid group, they have a LH to FSH ratio of 3:1. But with the electromagnetic radiation, that ratio it actually had dropped, which was a very good sign for us. Thirdly, we conducted an experiment in which we examined the effect of electromagnetic radiation on the hypothalamic pituitary ovarian axis. So you might say, okay, why the hypothalamic pituitary ovarian axis? Well, that is because it is the central regulator for the human reproduction system. So in your brain, just in brief, in your brain, you have a hypothalamus, you have accurate nucleus, and there are some tiny little neurons that secrete gonadotropin into your pituitary. And that pituitary now, it activates basophilic cells to help produce FSH and LH, which will then be released into your bloodstream via a feedback mechanism to tell your ovaries, here's what, this is how you are going to produce follicles. So with that, we were able to identify that in the accurate nucleus, we didn't see any changes at all. Electromagnetic radiation had no effect on the histoarchitecture of the neurons. In the pituitary, however, an estriol valvuloid model, there's a low number of basophils. But with electromagnetic radiation, there was an increase in the number of basophilic cells. In the ovary as well, we were able to see an increase in LH and FSH reactive cells. From this, we decided that we will investigate a post-irradiation effect. So since we saw differences, we decided, okay, you know what, let's remove that electromagnetic radiation and see what happens. When we removed that electromagnetic radiation, we were able to observe that the follicular cysts returned after 2 and 4 weeks. And we were able to see changes, which is like hypoplasia in the pituitary. At the end of this experiment, we were able to do a final analysis in which we investigated everything at the receptor, cellular, tissue, and hormonal level. And with this, now we were able to observe the electromagnetic radiation has no effect at all on our observable effect on our accurate nucleus. However, it does bring about changes in the pituitary in terms of the number of basophils and the morphology and also the ovary. In conclusion, we were able to, based on our results, we observed that 150 kilohertz electromagnetic radiation has some positive effects. It regulated the basophilic cells in the pituitary, potentially reduces the growth of follicular cysts in the ovary, but has no effect at all on the hypothalamus. This study, we would say, is not an exhaustive study. It still has a lot of limitations. However, because of the results that we have obtained, we believe that if persons continue to investigate the study, that this can be used as a treatment for PCOS later on. Thank you.